Hey, all you nature nuts. Welcome to the Outdoor Report, where we cover some juicy topics related to the outdoors. Today, we're going to go over 1,000 Hours Outside organization. We're going to cover how pine trees have a fun little shortcut. We're also going to cover a breakthrough in desalination. And we're going to go over a bird that popped up on my bird feeder that I've never seen before. So, without further ado, let's run the intro and dive right in.
Alrighty, so first up is 1,000 Hours Outside. This is a great looking organization that our board member Julia sent me the other day, and I was just looking it over. And you know what they do? The entire purpose of 1,000 Hours Outside is to attempt to match nature time with screen time. If kids can consume media through screens 12,000 hours a year on average, then the time is there, and at least some of it can and should be shifted towards a more productive and healthy outcome. So we kind of think adults can and should do that too, right? So I think everyone should check this out, because what they do is, they're motivating you outdoors, so you can track the hours that you spend outside, and they're just trying to motivate everyone to 1,000 hours throughout the calendar year. And you can download these sheets to get and to help yourself track and stay motivated and blah blah blah. See the free downloadable tracker sheets. If you head on over to their Facebook page, which you should definitely like and check out, they kind of keep you motivated. So one of the things I like that they posted the other day, where is it? Da da da. Sorry, here we go. So January launch ideas, like this is stuff that you can be doing throughout January. You get a first week of the year hike. I've already got a few in this year. This week, you should already, hopefully you've already got at least one under your belt. Milestone celebrations, right? Keep tracking of your hours. So if you got 10 hours outside, like, woo! Celebrate for every 10 hours that you do outside. That's every hour counts. Try to go somewhere new this January. Try to get outside to somewhere new. Maybe there's never been to a local area you've been dying to get to or whatnot. Recruit a friend. Socially distant excursions are pretty freaking awesome. And then try to up your hours. If you're out there for like a half hour or something, try to expand upon that. Try to get 2, 3, 4 hour days, maybe a whole day hike, overnighter, whatnot. And then besides recruiting a friend, they're saying pull together a small nature group. Here in Massachusetts, small groups are good. Socially distant, of course, mask and all that jazz. But that it gets us with people, around people, in wide, big, open spaces. So if you're feeling like you need to be with somebody outdoors, where it's at. So going back to their website here, if you got a family, this is a great thing for you to do in the year 2021 as a unit. You know, this is a child centric kind of program, but everybody can do this, and everybody should be doing this kind of thing, getting outdoors and getting after it. And if you got friends, if you got a family, and even if you got a nephew or nieces or nephews, it's always something to do and get them motivated and get you motivated to keep on getting outdoors. So we highly recommend you go check out 1,000 Hours Outside. That you can find them at 1,000hoursoutside.com. We'll leave a link in the description below, and also we'll link their Facebook page, because they have awesome, like, who, what, when, where, why, how, all that jazz. And you get locked into them, and they'll keep you motivated to get you outdoors. It's pretty awesome. And other than that, we'll be doing the same thing. But we thought we'd share this with you because we thought it was fantastic and something that everybody can do and be involved with, either with each other, by yourself, young, old. Everybody can do this. Everybody should be doing this. So um, it's fantastic. Go check them out. All right, next up is the story about how pine trees have a little shortcut. So, this is the fresh new year. Just had Christmas, just had the New Year's celebrations, and some of us still have Christmas on their minds. And I don't know about you, but when I was a kid, I was wondering how Christmas trees stayed green throughout the winter or throughout the time that we had it in our house as a Christmas tree. And you know what? Umeå University, probably didn't pronounce that right, but they're in Sweden. They found a way, or they kind of found the answer with that, and so they found that conifers can be green because of a photosynthetic shortcut, and how they actually figured it out is pretty interesting, and the fact that the conifers do this is also interesting. So I'm going to go ahead and read this for you. So the summary is: how can conifers that are used, for example, as Christmas trees keep their green needles over the boreal winter when most trees shed their leaves? Science has not provided a good answer to this question, but now an international team of scientists has deciphered that a shortcut in the photosynthetic machinery allows the needles of pine trees to stay green. So here's the full story here. So, um, we just read that. So, provided a good answer. Now, international team of scientists, starting right with that, including research from Umeå University in Sweden, has deciphered that a shortcut in the photosynthetic machinery allows the pine needles to stay green. The study was published in the Journal of Nature Communications, right? And this is from December 23rd, right? So and we're talking about ScienceDaily.com. I'll leave a link to this very summary, this article. Yeah, I'll leave a link of that below, so you can go and read this directly if you want. But or you can stay here and I'll read it to you right now. So in winter, light energy is absorbed by the green chlorophyll molecules, but cannot be utilized by the downstream reactions in the photosynthetic machinery, as freezing temperatures stop most biochemical reactions. This is especially a problem in the early spring when temperatures can be very low, but sunlight is already strong, and the excess light energy can damage the proteins of the photosynthetic machinery. The researchers showed that the photosynthetic apparatus is wired in a special way which allows pine needles to stay green all year long. Under normal conditions, the two photosystems, the two functional units where light energy is absorbed and converted into chemical energy, are kept apart from each other to prevent a shortcut and allow efficient photosynthesis. Right? So in winter, the structure of the thylakoid membrane, where the two photosystems are located, is reorganized, which brings the two photosystems in physical contact. The researchers showed that photosystem 2 donates energy directly to photosystem 1, and this shortcut mode protects the green chlorophyll in the needles when conditions become harsh. So, we have followed several pine trees growing in Umeå in northern Sweden over 3 seasons, says Pushanbag, PhD student. I totally probably got that wrong, but at the university, who has collected samples all around the year and made many of the analyses. It was essential that we could work on the needles straight from outdoors to prevent that they adjust to the higher temperatures in the lab environment before we analyze them, for example, with electron microscopy, which we use to visualize the structure of the thylakoid membrane. Imagine being able to look at this stuff under that kind of a microscope. That'd be so cool. All plants have safety valves to deal with the excess light energy, which is either dissipated as heat or as fluorescent light. However, only conifers seem to have such powerful valves that they can keep the photosynthetic apparatus intact over the extreme boreal winter. The research team combined biochemistry and ultra-fast fluorescence analysis, a very sophisticated method that can resolve chlorophyll fluorescence light at a picosecond time scale. I don't know what that is right now, but that sounds freaking small. Very small time frame. So that's awesome. Like this, they could demonstrate how the pine needles deal with excess light energy to protect their sensitive photosynthetic apparatus from damage. We need to adjust the equipment to study pine needles in cold temperatures in order to trap the unique mechanism, explains that person from that place in Amsterdam who has performed much of the ultra-fast fluorescence analysis. We also tried spruce needles, but they were hard to fit in a good way into equipment. Alfred Holsworth, sounds like a professor at Hogwarts, who has developed the time-resolved fluorescence measurements, adds, the pine needles gave us the opportunity to study the shortcut mechanism, also called spillover, as they really show an extreme adaptation. So this is like a real deal Holyfield stuff. Like, pine trees are badass. Conifers are badass. The study was done with pine trees, but the researchers believe that the mechanism is probably similar for other conifer species, like the typical Christmas trees, spruce and firs, found in where I am in New England. Because their photosynthetic apparatus is similar. This remarkable adaptation not only enjoys us during Christmas, but is in fact extremely important for mankind, says Professor Stephen Johnson from the university in Sweden. Hadn't conifers been able to survive in extreme harsh winter climates, vast areas in the northern hemisphere may not have been colonized, as conifers provided firewood, housing, and other necessities. Still today, they form the basis of the economy in most of the circumpolar taiga region. So that's incredible that they use this kind of technology to figure out, how our Christmas trees stay a little more green during the winter times. And as a kid, I wondered that, used to have that question. It's pretty interesting that right here, right now, 33 years later, we got this. And conifers play an extremely important role in our natural environment, and not just for humans, for everything else. And so now you know a little bit more about pine trees and their fun little shortcut that they have. All right, so our third story is the Desalination Breakthrough that I talked about. So the University of Texas at Austin found a desalination breakthrough that could lead to cheaper water filtration. So this is from December 31st, 2020. Producing clean water at a lower cost could be on the horizon for researchers after researchers solved a complex problem that has baffled scientists for decades until now. Right? So if you've been paying attention, our water, we're going to be needing to figure out something with our water systems, right? So it looks like the University of Texas at Austin has a cool new method for cheaper water filtration if we need to, or when we need to go that route. So producing, again, this is from ScienceDaily.com, and I will leave the link below. You can we hope you'll listen, but you can also just click on the link below and read the article yourself. But I'm going to read it to you. So, producing clean water at a lower cost could be on the horizon after researchers from the University of Texas at Austin and Penn State solved a complex problem that has baffled scientists for decades until now. Desalination membranes remove salt and other chemicals from water, a process critical to the health of society, cleaning billions of gallons of water for agriculture, energy production, and drinking. The idea seems simple. Push salty water through, and clean water comes out the other side. But it contains complex intricacies that scientists are still trying to understand. The research team, in partnership with DuPont Water Solutions, solved an important aspect of this mystery, opening the door to reduce costs of clean water production. Researchers determined desalination membranes are inconsistent in density and mass distribution, which can hold back their performance. Uniform density at the nanoscale is the key to increasing how much clean water these membranes can create. Reverse osmosis membranes are widely used for cleaning water, but there's still a lot we don't know about them, said Manish Kumar, an associate professor in the Department of Civil, Architectural, and Environmental Engineering at UT Austin, who co-led the research. And I'm sorry I got your name wrong. We couldn't really say how water moves through them, so all the improvements over the past 40 years have essentially been done in the dark. Findings were published today in Science. The paper documents an increase in efficiency in the membranes tested by 30 to 40%, meaning that they can clean more water while using significantly less energy, like 30 to 40% less more. less more. That could lead to increased access to clean water and lower water bills for individual homes and large users alike. We should send this to Flint. Reverse osmosis membranes work by applying pressure to the salty feed solution on one side. The minerals stay there while the water passes through. Although more efficient than non-membrane desalination processes, it still takes a large amount of energy, the researchers said. That's the downside. And improving the efficiency of the membranes could reduce that burden. Freshwater management is becoming a crucial challenge throughout the world, said Enrique Gomez, a professor of chemical engineering at Penn State who co-led the research. Shortages, droughts, with increasing severe weather patterns, it is expected this problem will become even more significant. It is critically important to have clean water availability, especially in low resource areas. So if you know anything about the climate crisis right now, you'll know why this is extremely important. The Science Foundation and DuPont, which makes numerous desalination products, funded the research. The seeds were planted when DuPont researchers found that thicker membranes were actually proving to be more permeable. This came as a surprise because the conventional knowledge was that thickness reduces how much water could flow through the membranes. The team connected with Dow Water Solutions, which is now part of DuPont. In 2015, a water summit Kumar organized. Remember, he was one of the co-researchers. And they were eager to solve this mystery. The research team, which also includes researchers from Iowa State University, developed 3D reconstructions of the nanoscale membrane structure using state-of-the-art electron microscopes at the Materials Characterization Lab of Penn State. Sounds super fun to be in. They modeled the path water takes through these membranes to predict how efficiently water could be cleaned based on structure. Greg Foss of the Texas Advanced Computing Center helped visualize these simulations, and most of the calculations were performed on Stampede 2, TACC's supercomputer. So, pretty incredible stuff when it comes to desalination, and thankfully we got this team working on it that we can hopefully get cheaper filtration to areas of disproportionate impact, because it's going to become an increasing problem very fast. And if we can work on it now, share the knowledge, get people on board, that would be a freaking miracle. And thank you, University of Texas, and all the other universities that are working on this, because this is extremely important. And just by having this simple little membrane, thicker, not thinner, can help be more efficient in desalination and doing all these things that we just talked about in the article. So, pretty amazing stuff right there. All right, our last topic is about that bird I told you that showed up at my bird feeder. So last year I got a brand new super cool bird feeder that I could totally impress my gram with my bird attracting abilities with. And yesterday one showed up, And I had something else planned for this, but this bird showed up on my bird feeder, and I was like, I've never seen it before. So I found out what it is, and here we are at the Mass Audubon Society. I'm in Massachusetts, so we're at the Mass Audubon Society, and I found out more about this bird, and so I wanted to share it with you. I'll put this in the link below so you can check out. It's part of a larger family, but we'll focus on this one bird. So this article was from a couple years ago, but it seems like it's still relevant today. So I'm going to read it real quick before I show you the bird. So it's titled An Epic Winter for Nomadic Finches. So as you can tell, we're already talking about finches. Every few winters, several bird species abandon their normal wintering areas to our northwest and move into Massachusetts by the thousands. While distantly related, redpolls, siskins, and grosbeaks all rely on food sources that go through boom and bust cycles, peaking and crashing every 3 to 6 years. And conifer and birch seeds, we just talked about them, right? Conifers at least. When conifers and birch seeds are scarce in Canada's boreal forests, these loosely related species erupt southwards in search of food. So, cool thing about this bird is it had to travel south to find us. And it's interesting because of how pretty it is. So the core group of these birds are collectively called winter finches. And this year will be huge for them. So this is 2018, so don't not related to this year, but also I think it relates at the same time because they're here. And they're here. There's a lot of them here. So we're talking about this guy right here, an evening grosbeak. So this year, these sunset yellow, black, and white patterned finches are the stars of the show. It's been a few years since Massachusetts saw any wintertime movement of evening grosbeaks into the state, and the last major eruption was in the 1990s. So this is even more like fascinating that this showed up at my bird feeder. Not just one, there is a group of them. So unlike many winter finches, evening grosbeaks seem equally happy feeding on several food types, both fruits and large seeds. They'll come to feeders, but their bulky size, which means they're burbs, they're plumpy, fat little guys, means that they prefer large platform feeders and will avoid tube feeders, which is funny because all of mine are tube feeders. Their fruit-eating tendencies means that they often move south with two other frugivores. Man, that sounds badass. Frugivores. I'm a freaking frugivore. Almost sound. Bohemian waxwings and pine grosbeaks, which may show up in smaller numbers this year. So, this is the bird that showed up, and it's been more common lately, right? Because since the 1990s, we haven't had a big boom like this. And I'm counting 2018 to this year the same because there's a ton of these birds in my neck of the woods, and it's right in the hot time for them. And again, they're supposedly don't like tube feeders, but all of mine are tube feeders, and there's a bunch of them. And it's been a few days since I noticed them, and now they're still here. And if you have a chance to see this bird, it is gorgeous. They look like they're having a blast out there. In terms of what I'm feeding them, they said they like fruits, and large seeds, but I'm giving them, pretty large seeds. I'm doing sunflowers, corn, the oil, and the regular bird seed that you can find at Ace Hardware because that's a co-op. So I like to support other co-ops. You should support other co-ops, and you should totally, if you're into doing the bird thing, like I'm really into birds, so if you can spot this one, let me know in the comments below. But other than that, I hope you learned something about this bird. Really beautiful bird. Well, that's a wrap for this outdoor report. Thank you all for being here. If you have any comments on all the stuff that we covered, please let us know below. If you have anything that has interested you, current events or anything related to the outdoors, let us know that too, and maybe we'll throw it up on the next one. Every Sunday, Here's an outdoor report covering current events that are environmentally focused. See you next time.